Good morning. My name is Laura Adang. I'm a physician scientist and child neurologist at the Children's Hospital of Philadelphia. It is my pleasure to talk to you today about BPAN, building hope for the future through research. And it is my joy to talk about some of the advances we have made together as a community over the last few years. While BPAN is a rare disease, it does not mean our voices are alone. There are over 7,000 rare diseases affecting 25 to 30 million Americans, and most of these rare diseases are actually single gene diseases, just like BPAN. Unfortunately, the vast majority of rare diseases do not have a treatment, and that's what we're going to join together as a community to help solve in the coming years. I want to open with, I am not alone, and the work I'm going to present is the work of many. We have a large group of people at the Children's Hospital of Philadelphia that are dedicated to pediatric rare disease research, but also dedicated to BPAN specifically. The foundation of our work comes out of the Leukodystrophy Center of Excellence, and I am a part of that group. But we also launched a dedicated clinic in 2021, and we were recently designated an NBAI Center of Clinical and Research Excellence, which is really quite an extraordinary honor. When we're talking specifically about CHOP BPAN, we have a focus on capturing what is most meaningful to families, and then figuring out how to measure that. We're interested in understanding the disease course because this is going to be essential for designing future clinical trials. And I'm most interested in exactly how it begins. How are we missing children in the medical system? How could we get them to specialist care and someday treatments faster? We're interested in developing biomarkers, which are things that you measure, and particularly biomarkers that can help us to understand or predict the future. We're also working towards therapy development. And I see these as all of the essential arms that will get us from where we are today as a community to potentially a treatment in the future. Our work is funded in combination by Don't Forget Morgan, as well as NIH funding through the Global Leukodystrophy Initiative. I'm going to give a brief overview of how we get a treatment from an idea all the way into patients. And it begins with two parallel sides. One is the patient side, where we're doing natural history studies and working together with patient advocacy groups to build patient registries so we know who the patients are. And simultaneous with that, we need discovery and research, getting potential ideas that we can test. And the goal is to bring those ideas together. My bench scientist friends help to do in the discovery and the research, and the natural history side, which is my area of interest, comes together to help us design the right clinical trial. And we're looking for both a safe drug, but also one that works and that benefit lasts a long time. And so that's one of the reasons the phase 4 study is so important. So after we go through the steps of a traditional clinical trial and have regulatory review and a drug gets approved by the FDA, we still need to monitor for a very long time. In the gene therapy world, it depends, but it's anywhere from 5 to 15 years we need to watch to make sure that the drugs stay safe, but also they stay beneficial. And my ultimate goal is to have these rare diseases added to newborn blood spots. But before we can do the testing in newborn babies who have no signs and symptoms, I need a treatment that can work. But again, while we're doing the natural history studies and the discovery and research phase, we should also be looking at biomarkers that potentially could be used on newborn screening cards. Because to me, that's the goal. The goal is to be able to both have a treatment, but also a way to detect BPAN earlier. And so here's one of my beautiful patients found by newborn screening that was able to receive gene therapy because of newborn screening. And that's where I want to someday be in the BPAN world as well. I think it's important to know that we can learn from trials that have come before us for rare diseases and avoid the missteps that have happened before. I think it's really important to know with therapy development that the treatment that you're developing is going to get into the cells that matter. If I can fix cell A, but the disease is coming from cell B, then the treatment will work in a cell culture, but it won't work in a patient. I think it's making sure that we are getting the medication into children in the right way. It's also important to understand how frequently the therapy should be given, when it should be given in the disease course, and how long it will take to work. There are things from a natural history study we can learn about the ideal target population, how to measure what at different time courses of disease. It might be that I might measure something different in young children than in an older population, right? I might need to design the trial very differently for those two different populations because they change in different ways. They're affected by the disease in different ways. But it's also really important to make sure that any trial we design has what we call generalizability. That means we have confidence that what we have done in a small group of children, sometimes clinical trials for rare diseases are 10 patients, 30 patients is not uncommon. But if I take that information, how much do I think it will help the whole population of BPAN? And those are really important questions that help to guide how we design clinical trials. We started off the process by asking parents to rank their priorities, to understand how BPAN affected children's day-to-day lives. And this work was led by a number of people, but I'd like to call out Francesco Gavazzi, who led this work, Emma Coates and Eric Yang, who are our clinical research coordinators that have been part of the BPAN clinic over the last 2 years, and Vanessa Smith, who's a genetic counseling student doing her master's work with us on this work. And what did they find? Well, parents told us what really is very intuitive. They wanted their children to have strong overall health, to be happy. They noted behavior concerns were a top concern of parents. They wanted better communication, meaning receptive and expressive language. Attending school was important to families. So was how the children ate and drank. And they also wanted independent movement, particularly indoors. And so while I probably would have guessed most of these, it's really important for us to ask families what's important to them, because it's going to be processes like this that will help us now match these health concepts to core concepts to build our future clinical trial. Next, Francesco asked parents to assess their children's adaptive behavior skills. And by this, parents said, how do children function in the real world? Because sometimes we can test something in clinic and it looks like a child does exceptionally well. But in the freedom of home life, that functional motor skill doesn't translate to actually being able to do something. And I will say even more often we see the flip, where children do much better in the adaptive world of home than they do in our clinic. And so I'm very interested in the Vineland Adaptive Behavior Scales, or VABs, because they correlate very strongly with our more traditional in-office assessments, but they can be done by phone at home. And they tell us what the child does in their day-to-day life, not just in the day where they came to clinic and they were really tired. And this looks at a variety of parameters, things from expressive and receptive language all the way to coping skills and gross motor and fine motor skills, right? And so we can start to parse out areas of strength or areas of need. And we can also look at how these skills change over time. And when designing a clinical trial, that's exceptionally important. The other thing we're doing, as I mentioned, is applying traditional outcome measures. The gross motor function, 88 or 66, are different ways for us to have a physical therapist capture function objectively. The positive is it is a very standardized objective function of what the child is doing in that moment. The huge minus is if a child is having a bad day or just doesn't want to participate, they won't score well on the score. And that's why we like to use a combination of both parental input as well as these more objective measures. We can also use things like the Peabody to measure grasping skills and visual motor integration, because these are things that are important for day-to-day life too. And we can capture overall skills. When are children with BPAN walking? When are they saying their first words? Are they having seizures, and how many seizures? And these are things that our group has undertaken over the last few years. One of our areas of interest, and this work is led by Anjana Sevagadamurthy with our retrospective natural history core, is to use medical records to fill in gaps, right? If a child is 2 years late to diagnosis, they are coming to me pretty late in their life, right? In a 4-year-old, I've missed 50% of that information. But parents are oftentimes in the medical system a lot with concerns. And whether the provider understands why the child is having, say, things like language delay or slowing of sitting, they're still documented in the medical records. And so we can, across entire consortiums or large groups, begin to collect medical records, collate, align them, collect MRIs, and collect the information from before we knew it was BPAN. And I will say this is probably some of the most important information we can gather, because this is the very early signs of BPAN that had been missed. And we can go back and use that medical records to help understand those early first moments. And so we've created this extraction process where we are assigning retrospective scores, so just looking at the medical records and saying, the child is walking with a little bit of difficulty, or things like that. And then we have data verification and PI approval and data locking. The goal is to make a regulatory ready data set so that someday we could use this information to inform a future clinical trial. I think it's important to understand why we need natural history studies. And there's some really potential and essential parts that a natural history study can give us. The first, as I alluded to before, natural history studies are absolutely essential for designing clinical trials capable of evaluating both efficacy, how well it works, and safety. We can also use potentially this information for things like non-concurrent control cohorts. So the traditional gold standard clinical trial is placebo-controlled. That means some children get the treatment and some children don't. In a progressive rare disease like BPAN, that just might not be feasible or even ethical to do. And so in those cases, what we do is we can use a well-done, both retrospective and prospective natural history studies to compare what is happening over the same course of time over children that are matched based on inclusion criteria, but this happened in the past, right? And so we've done this before for metachromatic leukodystrophy, where we took only retrospective records, meaning existing medical records, and we scored them in the same way they scored children that were in clinic as part of the clinical trial. And we compared how the children in our historical cohort compared to the children that were receiving treatment in that moment. It also can help us predict, outside of clinical trials, future outcomes. I want to someday be able to tell a family with a newborn, this is what I expect will happen in the next year, 5 years, 10 years, 20 years. I understand as a parent that is incredibly valuable information, and we will get there. The other thing it can do is it can help us to take our existing beautiful consensus guidelines and turn them into evidence-based guidelines. What are the things that are actually happening in the lives of our families? What are the things that we recommended that actually made a difference and helped? What have we missed because we weren't looking at the totality, right? And so things that evidence-based guidelines do is they can help us to really use real-world information to help improve clinical care for children, even outside of clinical trials. There are many ways that the research community has helped to build potential treatments or treatments to be tested for neurologic pediatric disorders. There are drug repurposing strategies where we take existing medications and we try them in BPAN animal models. There are also gene editing strategies, although those have not been able to successfully get into the brain yet. But a lot of our work at CHOP has to do with gene therapy. There are 3 different types of gene therapy that have been successfully used and have approved products for pediatric neurologic diseases. There's AAV gene therapy, where you directly make a virus, an AAV virus, and it contains the missing piece of the instruction manual. The advantage is that is off the shelf, meaning the same product can be used for everyone, so it is a faster process to get that into the patient. The big side effects is you're giving a very large dose of a virus, and so children have immune responses to it. Examples of this include the SMA, AAV9 gene therapy. There's another one for AADC, and there's also the Duchenne muscular dystrophy gene therapy. As far as lentiviral therapies go, the 2 leukodystrophies that have approved gene therapies, both are lentiviral based, both ALD and MLD. What that is it takes your cells, it removes them, it puts in the missing gene using a lentivirus vector, and then it puts it back into the child. The advantage is there's not the same immune response, right? Because you really did the virus part on the outside of the body and you're just giving someone their own cells back. But the big challenge for this is the manufacturing time. There's also potential risk of integration from the lentiviral vector. And so with the ALD product, there have been cases of myelodysplastic syndrome. And so that's something that you watch for long term. There are also RNA-based therapies, although I'm not sure that those would be relevant in the BPAN population. But the drug nusinersen for SMA is built using this strategy. One of the things I'm most excited about is the exploration of gene therapy for BPAN using optimized AAV capsids. AAV using naturally occurring capsids has been approved for several gene therapies, but there are some challenges. One is a pre-existing immune response, and second is that these capsids occurred in nature and they weren't designed to effectively and efficiently reach the cells of interest. And for a disease like BPAN, that's really important. And so what we can do, and what Dr. Beverly Davidson at the Children's Hospital of Philadelphia did, is she created a library of millions of novel capsid serotypes, and ultimately, through serial passage, identified an optimized pool of novel targeted AAV serotypes. And that means now you can match a optimized serotype to the areas of the brain and the cell types of the brain you are most interested in targeting. And so our hope is over the next 5 to 7 years, we're going to take the top performing capsid that reaches the areas of interest for BPAN and modify it to carry the WDO45 and test it in cell models and mouse models. And to do this work in parallel with a natural history study so we are ready for a clinical trial. So what are our next steps as a BPAN community? Well, we need biomarker development. We need therapy development and testing, and we need clinical trial protocol development, right? These are big tasks, but they're feasible. We can do this together as a community. I have no doubt. I'm incredibly hopeful for where we are today, especially compared to where we were 5 years ago. And so together we can solve anything, right? While diseases like BPAN are rare, we have loud voices and we have people that are motivated and interested in helping. So I am looking forward in the coming years to joining together as a community to help make the lives of our children better. Thank you again for your time. Again, my name is Laura Ading at the Children's Hospital of Philadelphia, and it was my pleasure speaking to you today.